Hello and welcome to Unit 2-1, so Lesson 1 of Unit 2 on DNA and genetics. Lesson 1 today is going to cover the structure of DNA. So to start with, let's go with a bigger picture, species. We as human beings are a specific species. And the definition of a species is any group of animals that can produce fertile, viable offspring. Okay? So fertile, viable offspring is the key to being a species. Now some animals, like a donkey and a horse, can have an offspring, but that offspring, called a mule, is not fertile and viable, meaning they cannot produce offspring. So why is it that a species has to have a fertile, viable offspring? Well, it's because DNA must match up. If DNA does not match up, then we get a lot of mutations. And when we have too many mutations, we have birth defects. And most of the time, things are not viable. So what is DNA? DNA stands for deoxyribonucleic acid, DNA. So deoxyribonucleic acid is present in all living things. You must have DNA to be considered alive. The basic unit of DNA, because DNA is made up of many, many billions of these nucleotides. So each individual building block of a DNA molecule is called a nucleotide. A nucleotide is the basic unit of DNA, and it is made up of three parts. The sugar, which is a 5-carbon sugar, 1, 2, 3, 4, and 5. And this specific sugar is deoxyribose. So deoxyribose sugar. Please make sure you know how to pronounce that. Deoxyribose sugar. It also has a phosphate group, and that is represented by this orange circle, and a nitrogen base. Okay? Now, these bases are what give it our DNA molecule, or our nucleotide. So the nucleotide, this is a picture of a nucleotide. And there are 4 different bases. And these bases give DNA its specificity. There are only 4 base pairs, and we'll get into that on this slide. The 4 base pairs you see in this DNA molecule are adenine, guanine, thymine, and cytosine. And as you can see, we have that phosphate group, and our 5-carbon sugar deoxyribose, and then our nitrogen base. And you can see here that the nitrogen base of 5-carbon sugar, phosphate, so this is also a nucleotide. Nucleotide paired to another nucleotide. Only thing different between them is the base. Each individual base gives it its specificity, okay, just as a review. And there's a specific rule that says what has to bind with what. So Chargraph's rule states that A's, or adenine, always bind to T, or thymine, and G's, which is guanine, always bind to C's, which is cytosine. A's always bind to thymine, G's always bind to cytosine. This can also be written this way: thymine equals adenine and cytosine equals G. Okay? It can go either way. But C's and G's will always be found together, and T's and A's and A and T's will always be found together. Okay? So whatever you need to do to memorize that A always goes with T and G always goes with C, that's what you need to know. That is Chargraph's rule. DNA is made up into one molecule, and it looks like a nice curly Q, and that is called a double helix. So what is a double helix? It is 2 strands of DNA that are bound together in the center based on the nitrogen base pairs. And again, as a quick review, A always goes with T, C always goes with G, and vice versa. So G always goes with C, T always goes with A. Helices are wound together tightly into coils, and then supracoils, and then eventually into chromosomes. And in a later lecture, we'll go into much greater detail on this subject. Chromosomes are tightly packed DNA created so that DNA can move efficiently during cell division and sexual reproduction. That is all chromosomes are. They are DNA, just very, very tightly wound DNA. And we'll show that again in these two topics will be done in a separate lecture. Human beings have 46 chromosomes, or 23 pairs of chromosomes. And the number 46 is important to us, okay? If you had a different number of chromosomes, you would be a different species. So it is important to know that humans specifically have 46 chromosomes, or 23 pairs of them. Our Geeks of the Week, we have Watson and Crick over here. And we have Dr. Rosalind Franklin here. And we'll get to those folks in just a second. Up until the year 1952, so prior to 1952, the scientific community believed that our hereditary information was carried by proteins. Okay? Up until 1952. In 1952, there was a series of experiments done by Drs. Hershey and Chase that proved that DNA was the hereditary material. All right? So before 1952 and their work, we believe that proteins carried our genetic material. After 1952's experiment, it proved that DNA was our hereditary carrier. In this time, there was only 3 labs in the world that were working on the structure of DNA. They tried to figure out, well, what does DNA actually look like? And the only lab in America was very small and very underfunded, and the head of the lab was Dr. Francis Crick. The second lab in England was run by Dr. Maurice Wilkins, and then there was a third. And their third one was run by Dr. Rosalind Franklin. And she died in 1958. And she was an X-ray crystallographer and a molecular biologist. She created the first picture, if you will, and specifically it's called an X-ray crystallograph for DNA. And this was her picture, done in 1953. Yes. The first time it was published was in 1953. And the problem with this is that there was no authorship given to Dr. Franklin. She passed away in 1958. Watson and Crick were then awarded the Nobel Prize for their work in the structure of it. You can see this is their double helix of DNA. And they received the Nobel Prize in 1962, 4 years after her death. So there's some controversy here because they really don't know how much credit Dr. Rosalind Franklin should have gotten for this Nobel Prize, because remember, she did it in 1958, or did it in 1953. She passed away in 1958. She took this picture in 1953 showing that double helix, which is what what Watson and Crick did a little bit later on. So the controversy is she produced the picture first, but there was no authorship given. She passed away, so they gave the credit to Watson and Crick, or so the controversy goes. And thank you guys so much for watching another edition of our video lecture series. If you have any questions, please feel free to contact me and we'll get those covered for you guys. And we will see you on the next lecture.